Good afternoon. It's my pleasure to follow the previous two presenters who kind of set the background, some regulatory issues, which I know is very near and dear to your heart here in the audience. I come from a different background. I'm from the drinking water industry. However, I have an interest in microbiology, which I understand most of you are also. So I'm going to give you an overview of this emerging, as we continue to figure it out, worldwide issue on harmful algal blooms, their toxins, and how they're affecting society. What I hope to do in my presentation here is give you an awareness of this issue and also some possible ways to mitigate it in your treatment facility, whether it's water or processing of foods. Here's a satellite view from NOAA of our Great Lakes in North America. They represent 1/5 of the fresh water in the world and 80% of the surface water that's available to us in North America. A very important resource. In this satellite view, the whitish gray areas is sediment reflecting light back. Moving down to the southernmost lake, which is Lake Erie, this real light spectrum is showing the green of a cyanobacteria bloom in Lake Erie. Lake Erie is the southernmost of the Great Lakes. It's the shallowest of the Great Lakes, and it's the warmest. And that allows us to be the most productive lake of the Great Lakes. There's many fish in Lake Erie as there are in all the other 4 lakes combined. It's also the most impacted by urbanization and agricultural runoff. And as a result, it's seeing the impact of harmful algal blooms and their toxins. Here's a little bit closer up shot of a late summer Microcystis bloom that typically starts in the western basin, which is separated by islands there, and then moves into the central basin. Once again, the lightish blue area is sediment. The darker green color are the cyanobacteria that, in this case, is blooming. The whole south shore of Ohio, from Toledo past Cleveland, is pretty much impacted by this bloom. This is a particularly bad one. It doesn't happen this severe every year, but the potential is obviously there, and it's not necessarily getting any better. I'm affiliated with Ohio State University, and they have a biological field station called Stone Lab. It's shown on this diagram up here in the middle of Lake Erie on one of the islands. It has to be perfectly situated to study harmful algal blooms based on this picture here, as you can tell. Unfortunately, Lake Erie is not the only problem we have in Ohio. Our inland lakes, of which my title slide is a shot from our largest inland lake called Grand Lake St. Mary's, it was the first, and it's still the most severe impacted by these cyanotoxin blooms. This is a bloom that's on its way out, comes to the surface, and you get this very, what I call Monet-like scene. I think it's quite pretty, actually. But in addition to our Great Lake and our inland lakes, last year we saw something else develop, and that's the Ohio River to our south boundary. There was a 600-mile bloom, 600-mile long bloom on the Ohio River. All these sources, the Ohio River, Lake Erie, and our inland lakes, have water supply intakes on them. Some are significant cities like Cincinnati, Toledo, and Cleveland. So I guess I'd like to say I'm from Ohio, and we have a horrible algal bloom problem and a cyanotoxin problem, and we've reacted to it. I hope to share some of that, what we've learned with you. Having Toledo happen, there was a large public outcry, a large press feature, whether you lived in the state or not, you heard about it. And our regulatory agencies have appropriately reacted and come up with probably stronger monitoring and regulatory requirements for the water industry than most other states or other countries have done so far. So what I hope to cover here are 4 particular areas, touch on them. First step is, in mitigation, is to realize whether you have a problem or not. And that's source water detection. So I'll cover that a little bit. Then once you think you may have a problem, you need to define that problem through some analytical testing in the lab. And then once you have the problem, how are you going to take care of it or prevent it in the future? And then I'll finish up with some potential sources of risk to the food industry and the drinking water industry that we've already heard from the previous speakers. Source water detection. One of the first things that you'll see is a bloom. And blooms are pretty obvious in most cases because of the fact that they come to the surface. However, a visual bloom does not mean you have a problem. It's a warning sign. Not all blooms are cyanobacteria. Green algae or diatoms, etc., can cause blooms that don't necessarily have toxins associated with them in fresh water. Not all cyanotoxin blooms produce toxins. So because you have a bloom doesn't mean you have toxins. Not all blooms are visible. Planktothrix, which is one of the organisms we see in the Great Lakes, doesn't necessarily form a bloom, but it does form toxins. So on the left we have a bloom that doesn't have toxins. On the right we have a bloom you can't see, but there are toxins. And fourth, you can have toxins without a bloom because the bloom may have disintegrated, settled out a couple days ago, and toxins persist in the water column for days to weeks to, in some cases, months. So a bloom is just an initial warning that you should do some more investigation. If you have a large lake, there's satellite potentially can identify you to potential awareness that there's a bloom occurring. On the left is a real light spectrum. You can see the green around the island areas of Lake Erie. And then if you do spectral analysis, which NOAA does through their satellites, and you look for the pigments that are specific to the blue-green algae, cyanobacteria, phycocyan in this case, you can come up with a concentration, the darker the color, the redder the color, the higher concentration, of the algal cells. Not the toxins, but the cells. It's another tool that's available to tell whether you might want to do further investigations. Once you have a bloom, then you want to find out what's in the bloom. Is it really a cyanobacteria or not? And the old microscopic method is a pretty foolproof way to do that. There's 5 particular species shown here that we see in the Midwest area. And you can do that through just taking a grab sample if it's a bloom, or using a plankton net to do a concentrated sample. And then you can ID it to the genus and get some counts. Remember, I think Leslie just showed enumeration of so many per ml is a warning sign. If you get into the 6-7 figure digit, there's a potential for toxins that may be a problem. There's also some automated methods. FlowCam is one of those. It's based on particle imaging, and it can be trained to recognize particular Microcystis colony, for example, count it, and let you know how much you got concentration-wise and what the identification is. Another indirect way to see if you have a bloom going on is water quality parameters. Since these are photosynthetic organisms, several things happen during a bloom. All the parameters listed there increase. The pH increases. The pigments that produce the photosynthesis, the chlorophyll A and the phycocyan, and in the blue-green case. Turbidity, conductivity, dissolved oxygen has a diurnal peak during the day. And temperature is an important parameter too. You can do this with a one-time grab sample, go back to the lab and measure the pH, but what we're doing in the Great Lakes is we have these continuous data sondes, and a sonde is nothing more than a group of probes put together on a single device, and then it's hung from a buoy. Sometimes you have them at different depths, and that information is reported back either by cell phone or satellite or some other method, and you get real-time data, as shown over here on the right-hand side, of what's going on. Toledo, several years ago, had an event that came upon them very quickly, and it happened to be on the weekend too. The wind shifted, and they didn't have these real-time data. This may have made a difference if they have it. Now there's a couple dozen of these buoys at that website shown up there throughout the Great Lakes area for advanced warning signs. Taste and odor was mentioned as not a good parameter. However, it's another good warning sign. The cyanobacteria do produce the taste and odor compounds geosmin and 2-MIB that produce the taste in drinking water. But other organisms do that too. So if you have a taste and odor event, you can't assume you've got cyanobacteria. Not all cyanobacteria blooms produce taste and odors, and at the same time produce toxins. So you can have a bloom with taste and odor or without taste and odor. It's another warning sign that you might want to do further investigation into your water supply. Analytical methods. I'm not an analytical lab person myself. This is what my research has shown. The standard methods are still evolving. There is no perfect method yet, in my opinion, for the microcystin or other toxins. But there are various options available depending on whether you want to do some field monitoring or you want to do specific identification of which variant or conjugator you have of the microcystin. One of the problems with the microcystin that was mentioned, there's 100+ different variants or conjugators of that toxin. And there's only about 6 or so standards which come into play when you're using the mass spectrometry. You need some standards to come up with valuable results. Some of the methods also have a reporting level that's very close to the health advisory, which can cause problems with regulatory issues. First, before you analyze, you need the sample. It's very important to use glass or PETG bottles. Regular plastic will absorb the toxin and give you false negatives. If you're using tap water, sampling tap water in your processing facility or water plant, you need a sequestering agent for the oxidants that may be in the water, chlorine particularly, normally. Samples need to be refrigerated or frozen depending on your holding time. And then you need to make a decision on how you want to prepare the sample. As we heard, the toxins are often held within inside the cell. If you just want to look at what's outside the cell, you need to go through a filtration process. If you want to look inside the cell, you need to go through a lysing process. And there's several ways to lyse: chemical sonification and freeze-thaw. In Ohio, our standard method is freeze-thaw. That process in itself takes some time because you've got to freeze a sample. We use 3 times and thaw it out 3 times, which can actually take a day's worth of work in the lab. And then the next day you end up processing the sample. What most people look at is the total toxin load from a raw water sample, a lake sample. So they do a sonification or a lysing process to include both the internal and external toxins. Important concept to remember when you're sampling and analyzing. This is my synopsis of the 4 common chemical methods, analytical methods that are out there. Enzyme link immunosorbent assay, ELISA, several different ways to utilize that technique. High pressure liquid chromatography followed by UV, or high pressure liquid chromatography followed by mass spec in tandem. And then the qPCR, which I understand, having visited the exhibit hall down here, is a method that you use in the food industry. We're just now learning about it in the water industry. It's not that available to us in Ohio at this point in time. All right, a little bit on ELISA. For microcystin, we use the ADA method, and the ADA method allows us to detect the total microcystins, not just LR, of the 100 or so variants that are out there. Shown on the diagram here is a microcystin molecule. Every microcystin has this ADA group shown in the rectangle. The difference between the different variants is what the amino acids they have that are attached to the various points on the molecule. If we can disregard that and go with the ADA group, we get a real good indication of what the total microcystin content is of the sample. We cannot identify the variants, but in many cases, that's not our first concern. We want to know what the total potential toxin level is. It's an indirect method using an antigen, of an antigen using an antibody. It can be used both on natural samples and tap samples, drinking water samples. There's no concentration step needed. It's sold in kits. There's really no high-end equipment needed or expertise. Pipetting is the key, so you do need some lab technique. The technique is certified by US EPA, and it's certified in Ohio. It's relatively inexpensive. One of the drawbacks is the quantification is based on the variant Microcystin LR, and the toxicity of some of the other variants isn't quite figured out yet, so it can under or over-report some of the variants. ELISA comes in in the lab. This is what we use in Ohio. This is a plate kit. You can run multiple samples at a time and generate a standard curve every time you run a sample. The samples are in these reaction tubes. It's read by a plate reader, and the color is inversely proportional to the level of the toxin, microcystin in this case. The range is between 0.15 and 5 parts per billion. In Ohio, the reporting level is 0.3 micrograms per liter, which is right where health effects is for the younger part of the population. Relatively quick. A lot of the time is taken for reaction time. In Ohio, we find it's meeting our operational needs as far as drinking water facilities are concerned. We can do it in-house. There's also kits available for the other toxins shown there. There's also what I call a field-based test strip. This is a good screening method. Takes about 30 minutes if you want to do your lysing, which is highly recommended on natural samples. We're up to about an hour to run the test. There's 2 ranges available, 0 to 5, 0 to 10. And then for the 3 toxins shown there, there's strips that are available. Each strip has a control line on it. And then again, it's an inverse color reaction. So the highest level has, in the second strip here, has no color. This is the control strip. And then the lower levels, the control meets the test. There's also a lab-based test tube method, another good screening method that's a little bit more quantitative than the strip. Still need to do lysing, still need to generate a standard curve like you do in the plate kit, and you measure the absorbance on a photometer. These results aren't good for regulatory use in the drinking water industry, but gives you a good operational parameter. Moving on to high pressure liquid chromatography. The chromatography separates the compounds, and then the absorbance at the UV level of 238 is measured. This is a non-selective, and there's other co-eluting interferences that can be detected. It's less expensive than the next method using mass spectrometry, and its level of sensitivity is on the same order of what the ELISA kit is. Liquid chromatography with tandem mass spectrometry. This often requires a solid phase extraction step, which can cause some interferences. It's not suitable for natural waters. The sensitivity drops now an order of magnitude lower. The variants can be ID'd with the microcystin, whether it's YR or RR, but you need standards to do that, and the standards are lacking for many of the 100 or so variants. It's more expensive than ELISA and the LCUV, and it takes a highly skilled analyst and equipment. There are standard methods, US EPA 544 for microcystin and 545 for anatoxin A and cyalindosporopsin. The US EPA 544 method is limited to the 6 microcystin variants that we have standards for, and this is the method that they're going to be using for the UCMR. Something that's relatively new to me is that the LCMS method can be enhanced by using this 2-methyl 3-methyl oxide 4-phenylbutyric acid, which is MNPB, which chemically cleaves the eta group off, and then you measure the eta group, so it gives you a total microcystin count, including all the variants, much like ELISA. You can use it on natural waters as well as tap water. You don't need to go through a lysing process. Relatively quick, sensitive, and it lines up well with ELISA as far as the results being confirmed. Polymerase chain reaction, qPCR. Genetic-based technology that simultaneously can quantify the total cyanobacteria present, along with the genes responsible for toxin production. The total cyanobacteria is determined by the 16S rDNA gene, and those counts correlate well with cell counts. Second part of this identifies the genes that produce the toxins, the 3 listed there. It's relatively quick. In Ohio, it's very limited in its availability. There's one facility that does it, and it's Ohio EPA. So it's not something that's generally available to us in the drinking water industry. What's nice about it, it's very specific. If there is no gene, there's no toxin. However, if there's a toxin gene, doesn't necessarily mean it's been turned on and producing a toxin. It's a very proven method in the diagnostic field, very sensitive, high sample throughput, but it's costly, and there can be some inhibitors. There's a reference website down there. In Ohio, the Ohio EPA did a study of our natural waters in the state, and they found 16 microcystin variants. The most common are listed there. The YR was more common than the LR, and the microcystin RR was the 3rd most common. 91% of our samples had a microcystin variant that's not detected by the US EPA method 544, which is the LCMS method. Some of the regulators in Ohio feel that the LCMS method will underreport the total microcystin because of the lack of standards. But the addition of the MMPB, cleaving of the eta group, agrees very well with the ELISA method, and this may have potential in the future. So now we've seen our blooms, analyzed our toxins. What do we do to mitigate the situation? Well, the best mitigation is prevention. Nutrients are causing these blooms to occur, both internal and external. Internal is runoff from point sources and non-point sources. Internal is from phosphorus being released from the sediment when the lakes go anoxic. The phosphate comes unbound from the sediment and becomes available. So controlling input and also mixing, as I believe Leslie talked about, can disrupt the formation of blooms and keep the bottom layer from going anoxic. If you have a source water, many drinking water facilities monitor for algae on a weekly basis, so they get cell counts and ID. If you see a particular alga increasing to a bloom status, we can hit it with an algicide. Copper sulfate is a common one, before a bloom gets established. What we don't want to do is to put an algicide on a bloom that's well into the 100,000 cells per ml, because now you're going to release all the toxins that are in the cell, and you're going to have a huge problem at your treatment facility. Many facilities have a way to avoid a bloom, going to a different source of water, different reservoir, different upground reservoir, or manipulating the intake depth. If you have a large lake, oftentimes, or all the time, the blooms are at the surface, and you can avoid if you go to a deeper depth. Treatment options. First thing you want to do is get the cells out intact. As we heard, most of the toxins are intercellular, and we don't want to disrupt the cells, lyse them during the process. So many water plants, surface water plants, are set up with the method to do that. That's the principle of water treatment design, through coagulation, which destabilizes the particles, flocculation, which builds the flock, settling, clarification, which settles out the particles, and then filtration through either sand filters or low pressure membranes, micro or ultra membrane filtration. Does a great job of taking the cells out if you don't do it too violently and disrupt or lyse the cells. Once you go through that step, then there's several processes available to remove the toxins. Reverse osmosis is a fairly common method. Oxidation is the key in many cases, because every water plant does oxidation through some sort of disinfection. I think every processing plant has some sort of disinfection process. Ozone's the best. Free chlorine is commonly used in many drinking water facilities. Free chlorine is most destructive of the toxins at the lower pH. Potassium or sodium permanganate is also used. And you can use UV if you have very high doses and you add hydrogen peroxide with it. In all these cases, the contact time is important, as is the dose. Most drinking water facilities, the dose of the chlorine and the contact time for disinfection is on the same order of magnitude you need to knock down a small level of microcystins in your source water. So that's the good news. This is a table for reference on the effectiveness of oxidants. Along the top are the four main toxins, and then on the left-hand side are the oxidants that are typically used. The yellow boxes are good, indicating that chlorine takes care of three of the four toxins. As you can see, none of the oxidants has yellow all the way across, so there's no perfect scenario for every toxin that's out there. Another step is absorption through activated carbon. This is a facility probably available to many food processing facilities. On the left is a large pressure filters full of carbon. That happens to be at my particular water plant, put in for multiple reasons. Toxins is one of them. And then on the right is a more lab or smaller process parameter for taking care of toxins or potential toxins if they show up in your municipal water and you got to treat it. The GAC is a little different than the chlorine. It's a little more absorbent at the higher pH. And of course, the contact time, empty bed contact time is important here too for the absorption process. So what we need to do in the water supply business is to optimize what we have. Our current treatment processes can do a pretty good job if you get them optimized. You need to have the right chemical feed capacity. Toledo, in response to their do not drink, was feeding powder activated carbon, but they were limited in the quantity they could feed. Over the winter, they doubled their quantity, and they were able to deal with the large bloom in 15. So you got to have the right capacity, you got to have it optimized, and you got to stop any recycling from your filter backwash or from your settling basins. Multiple berries is the key in drinking water. Source water options is good. You want to remove the cells intact. Absorption, and then finally the oxidant. And the dose and contact time is key. Most facilities don't have a whole lot of contact time, but you can build that in by having a higher dosage, and it's distributed throughout your distribution system. The contact time continues to build. Final few slides is information we've kind of heard already on what potential impacts might these cyanotoxins have on foods. Fish and shellfish. Shellfish has been an issue in the marine environment for a long time with algal toxins. We've heard that organs is a primary target. However, it does show up in mussels. At Ohio State University, Sea Grant group has done some research just recently on sport fishery, which for us is walleye and perch. And they do see, in some cases, during high blooms, the total daily intake being exceeded in the mussel. As we heard already, heat doesn't destroy the toxins. Livestock, we've also heard about that. There's definitely been proven deaths of livestock and dogs. However, the feed and the forage and then the milk and the meat is an item that's still up for research. Crop irrigation, I'll touch on next. Processed water at your particular facility. If you can have activated carbon of some sort or chlorine, that would help mitigate. In-store misting, I don't know how many people are aware of that, but when I buy my produce, it's kept moist, so somebody's using some water to do that, and that's a potential source if you're not paying attention if it's a municipal source. And my last one is supplements. This is crazy, but this is probably not regular. This is the least regulated of everything that we do. And some of this blue-green algae you buy in health food stores comes right out of a lake, and the testing, in my opinion, is minimal. All right, irrigation. There's both spray irrigation and ground irrigation. They found toxins and algal cells in leafy crops, particularly lettuce. After 10 days, there's still been a viable algal cell there that produces toxins, and the toxins aren't easily, and the cells aren't easily removed. Ground irrigation, we heard some already about uptake for roots. A lot of this research is in its infancy. A lot of it's been done with hydroponic work, so equating to the field is still a research item. I list some areas there where microcystins have been found in crops. The toxins also affect the plants. Reduce seed germination, seedling growth, and quantity of the yield and quality of the yield. And there's some bioaccumulation. My concluding slide is, we want to mitigate, we need to monitor. Is there a potential problem? We want to screen for the toxins. They can't occur year-round. We want to test using some analytical methods. In Ohio, we currently are hanging our hat on the ELISA ADA method, and they're checking, and the Ohio EPA is using qPCR to see how the two correlate. It's a research project, information collection that we're doing in Ohio. Treatment, the good news is there's treatment available to reduce toxins depending on what you're starting with, the level in your water. You want to take the cells out intact. Free chlorine does a great job. Ozone's great. I know some of you have ozone in your facilities. Activated carbon's good too, but each one has to be dealt on a case-by-case basis. So that concludes our presentation. I think we got some time for questions for myself or the group.
Hi, thanks for a great talk. And as a citizen of the Columbus metropolitan area, it's good to see that we have so much good work going on near us. So, you know, it seems to me that the whole field of cyanotoxins is still pretty early. And you mentioned that there's hundreds of them, and right now there's not analytical methods for a whole lot of them yet. So it seems like, at least in the past, in other areas of food safety, what typically happens is that people find a method that works for a toxin, then everybody starts looking at that. So has there been any talk of having maybe the alternative approach where we could look at all the cyanotoxins, prioritize from a toxicology risk perspective, which ones are actually probably the most important, and then use that to help guide which methods should be developed? Because I think even then there's a knock-on effect of you could then also determine what is the analytical limit you need, because you may find that cheap and dirty methods are could be effective enough to find the level that presents a risk. You don't need an LCMS MS method if you need 5 kilograms to kill somebody. So has there been any discussion of kind of looking at these things more broadly using structure-activity relationship or anything?
I'm not the methods person, but I like information that can be used quickly in a process, whether it's drinking water or food. So not something you need to send out a lab and get results back several days later. Leslie?
Thank you. As part of the Interagency Working Group for Habarca, we have a group, a subgroup that some members are FDA, USDA, EPA, and others that are working only on methods. And I think that hopefully experts and chemists and all these people that are part of the work group will come up with something like that, hopefully soon. But they are working on that right now.
Okay, thanks.
Yes, we do hospital and dental university water systems globally, and we do a lot of testing of municipal water supplies, and we find that oftentimes they have very elevated pHs. Some municipalities add phosphates. They're trying to grow biofilms to protect against, I guess, leaching of various minerals and such from their infrastructure. And yet, in your slides there, I was noticing that you had a pH of 7, or you had neutral pH for water in Toledo. Is that the way you distribute it as well, or is there a further treatment afterwards that elevates pH and perhaps adds phosphates?
pH in drinking water, I didn't want to say that Toledo's pH was 7. What I was trying to indicate in that slide is that the lower pH is more effective for chlorine, free chlorine, to destroy the toxins and also for disinfection purposes. pH of drinking water can vary depending on the process used to treat it. Some places use lime softening that jacks the pH up. Typically, at my facility, we use lime softening, but we bring it back down with recarbonation, which is CO2, carbonic acid, to bring it back down. And we try to leave between a 7 and 8 pH at my particular facility or in Columbus's facility. And we do use phosphate to provide a coating for lead and copper corrosion control. Toledo's does do lime softening. I'm not sure what their pH is.
Some of the values we've seen are up to 9, 9.4. This is a pretty serious concern when you're using source water chemistry, whether you're adding a surfactant for a cleaning process potentially or a disinfectant, and you're expecting a specific pH. So anyway, I'm just bringing that to light. Thank you.
You mentioned source water treatment methods. I know there's been a little bit of work done on alum as a way to treat your source water. What has Ohio's experience been with that?
Ohio's largest inland lake, Grand Lake St. Mary's, has been a problem for harmful algal blooms for a while. And they did a partial part of the lake treatment with alum, aluminum sulfate, spread on barges. And it was a multi-million-dollar effort that I'm not so sure paid dividends. The way that the process works is the alum kind of coagulates the phosphorus and ties it up in the sediment. This particular lake is 5 to 7 feet deep, and there's a lot of wave resuspension, and it's a large lake, and they only did part of it. But that is a proven method to bind up the nutrients, phosphorus. I think you're much better off controlling what comes in and keeping the bottom from going anoxic.
Just kind of want to make a comment. I'm an analytical chemist that is currently working on the validation for USMR4 with the EPA for a laboratory. And one of the things that we are doing is trying to use a combination of these methods for the actual finding of these test samples, where you're starting with the ELISA, ELISA, in order to kind of get a screen of it, and then using the LCMS methods to try to find of those 6, specific ones. It was an interesting slide that you said that 91% of the samples that was taken do not have those 6 analytes in question or microcystins in question that we're looking for. So it is interesting for that MMPB method. It would have been nice to kind of see that implemented in the UCMR4 criteria, because I think it would give us kind of a better number of tests that we could utilize for the current UCMR4 criteria.
Well, as we've heard, there's no perfect method, and the methods are still evolving. Something's got to be chosen to move forward with. There's a lot of constraints there.